Thank you. So I'm Bennett Levitan. I'm a Senior Director at Janssen R&D, and I'll be discussing the third component of the framework on the application of preference data to inform regulatory decisions. So we go to the next slide. There are lots of ways you can use preference data to support a decision. Sometimes it's kind of straightforward, but it often gets messy because there's so many things you could do. So component 3 of the framework gives you some structure for applying preference data to decisions. Next slide. So the way we think about it is there are sort of 3 broad classes of techniques by which you can apply preference information to support a decision. You could use the preference information alone. You could use the preference information and clinical data viewed jointly, or you could take the preference and clinical data and mathematically combine it into what's often called quantitative benefit risk assessment. And one example of each might be the relative importance of the attributes, which you get from the preference survey alone. The second is the graphical techniques, where you might look at preference and clinical data together. And the third might be a choice share. So next slide. We'll look at the preferences alone. So here's one example. I'm going to show you a series of examples because they make it easy to follow how they could be used. So consider the disease Fragile X syndrome. It's a rare genetic condition. You've got learning and intellectual disabilities and cognitive impairment, and there's really no cure. So a company did a preference study during phase 2, because in this rare disease, people had no idea what the real endpoint should be, and they wanted to prepare for what endpoint should be used in phase 3. Next slide. So there are 6 attributes, like learning and applying new skills and explaining needs. These are explained more obviously in the survey. Now animate, and when the people conducted the survey, next slide, Carl, there were 2 attributes that were considered most, Carl, go back please, that were considered most important. Prior slide, please, Carl. The first and the fourth are the ones that the clinical and commercial people thought were the most important. Now, the size of the purple line shows the relative importance of the attributes. And you could see the fourth one is actually the least important. So the clinical and commercial perspective actually got it totally wrong. The caretakers, and the preference study was done on the caretakers or the parents because the children couldn't do the survey, actually regarded controlling your own behavior and caring for your own self as the most important attributes. So it shows you that you can use a quantitative preference survey and get radically surprised by what people think is the most important thing that you should study or what your drug should do. Now, as it happened, this drug failed in phase 2. Had it not, the company would have used this information to design the particular endpoints that they would use in phase 3. So I think it's a fascinating example of how off our instinct can be. Next slide. So that was relative importance. Now, a very common tool that's used, or metric that's assessed from preference studies when you're using the preference study by itself is maximum acceptable risk, or MAR. This is an old example, but I always like it because of how surprising it can be. So this is in Alzheimer's, and people were asking if I convert 1 year of moderate symptoms to mild symptoms, or 5 years of mild symptoms to moderate symptoms, to lesser symptoms, how much risk of death or severe disability due to stroke would you be willing to accept? So people are terrified of Alzheimer's, so these lines are pretty high, these bars. So if I were to take 1 year of moderate symptoms and make them only mild, people would take up to a 5% chance of death or permanent severe disability due to a stroke. Now, if you essentially halt the disease, which is a 7-year time frame in the study, and keep your symptoms at the mild level, people would take over a 30% chance of death or permanent disability. Now, that's insanely high. And what this is telling us is, 1, people are terrified of Alzheimer's, and 2, people would probably be willing to accept risks that are beyond anything that any company or health authority would be comfortable putting on the market. Then, when you do this type of information, it has very good roles for benefit risk, but it also has roles in assessing a target product profile. What are the ingredients for a successful product, or what are the thresholds that are acceptable? So that's another example of a way of using a preference study by itself to gain information for a decision. Next slide. So what if there's more than one risk? So that was a maximum acceptable risk for just one risk. Well, this is a technique that's becoming more popular. It's still very new, where you could look at multiple risks versus a given benefit. So here it's rather abstract. If I take people from severe to mild symptoms, I have risk 1, adverse event 1, and adverse event 2. Anything below the red line is acceptable. In other words, any combination of two risks that's below that line has benefits exceeding risk on average. The maximum acceptable risk for one of the adverse events by itself is on the axis, so a little over 8% for adverse event 1, a little over 5 for adverse event 2. Next slide. Now, if you don't know the degree of benefit, or there are multiple degrees of benefit, which is always the case, you can apply the same idea. But now, if you have a less valuable benefit, like only moderate to mild, you still have a viable product where benefits can still exceed risk as long as you're below the yellow line. Next slide. Now, we were just looking at the line, but that's sort of the average preferences. What if you want to take into account the population heterogeneity of preferences? So around each line is a distribution. And the red line is, say, the mean preference, but the blue line gives you the distribution of the maximum acceptable risk, where 90% of your sample would choose to accept the combination of risks. So this is a way you can begin to not just think about two adverse events or multiple adverse events versus a benefit at once, but also to what degree population heterogeneity interplays with your preference assessment. Next slide. Now, these are all linear trade-off curves. There's no reason they could be constrained that way. I was just doing it to be simplistic. But in the real world, models can be just like this and much more complex. And we're only depicting this in 2 dimensions, but there are tools that have been developed. In fact, the FDA used one for an obesity device approval back in 2015 that can do essentially the same thing as this curve. So that's another way you can use preference information alone to support decision making. The last one I'll show starts on the next slide. So this is a mock example. It actually is from the obesity case study, but I'm just going to use it to build a story. Let's make believe we have a new efficacy endpoint, the time to an event. And I would like to know an effect size, that's meaningful and I could use to feed my statistical hypothesis test. But I have no idea. It's a new endpoint. But I do know an effect size that I trust, say, for heart attack. So I can use a preference study to connect these and come up with an effect size for the new endpoint. So next slide, or animate, Carl. So I start with a well-accepted effect size. I just made it up of 0.5 for heart attack. Animate. So now I ask, what is the preference change that's associated with that accepted effect size? That's the black double-headed arrow. Animate. I now move that over to the novel endpoint and animate. I take that down to the x-axis, and I get the preferentially equivalent effect size. So in this mock example, a change of 2 years in this novel time to efficacy endpoint is preferentially equivalent to the well-accepted effect size of 0.5% for heart attack risk. Again, these are made-up numbers, but the point is, if people trust the effect size for the heart attack risk, I can use that same trust for a now 2-year effect size for this novel time to efficacy endpoint. Now, this is certainly not the only way to come up with effect sizes, but it's one way you could use preference studies to inform them. Next slide. So those were methods of using preferences alone to inform a decision. Now, when you start bringing in the clinical data, you can start looking at this information together. And typically, these are graphical techniques. So next slide. So here's a forest plot in an anticoagulant with mock data. So each row is a different endpoint. The 34.2, the minus 34.2 means if you have 10,000 patients treated for a year, there'd be 34.2 deaths due to any cause on the study drug versus the comparator. So anything on the left favors the study drug. The endpoints are in order of impact or preference, essentially. The most severe ones on top, the least severe ones on bottom. And you could see anything on the top and the middle either favored the study drug or on the zero line. It's only the things on bottom that favor the comparator. And this is actually very similar to a slide that was used at an advisory committee meeting with the FDA. And you didn't have to take it any further. It was obvious that the study drug was helping where it was important and hurting where it was less important. Now, in this particular slide, for simplicity, the endpoints are listed in order of preference rank. Next slide, you can certainly take this a step further and displace them by their actual preferences. For example, you could see the large gap between disabling stroke and everything on bottom, which is pretty important to the benefit risk story. And this, in theory, is a good idea. In practice, it can get awkward because the endpoints will land on top of one another, and people will start questioning the data more than they would just rank. So it's fine, but you have to ask whether this is the additional complexity of this type of visualization is worth the benefit. And if this is too messy, next slide, you can go into tabular form. So this is essentially an effects table with the two studies, a rate difference, a graph of the rate difference, and a graph and numbers for the preference weight. And then you could mentally decide how to balance these using the rate differences and the weights, or use your judgment whether you trust the weights. It's a very useful summary. It basically is an augmented effects table. Now, when you look at this, it also makes you think, well, why can't we go the next step and combine that information, the rate difference information, and the preference weight information? And that brings us into the third class of methods. So next slide. So here we're talking about the quantitative benefit risk methods where you mathematically combine preference and clinical data. And I'll just do two methods. There are a variety of techniques around, and they're growing in popularity, and they're beginning to become less academic and more used in the regulatory context. The first one, next slide, is called choice share. And this actually was used in a regulatory decision by CDRH. You're asking what proportion of patients would use the treatment. So this was for a medical device. And in this case, it's will I choose to use the device or not? And there are various properties of the device listed in the boxes. And then on the right is the percentage of people who would choose to use it. So virtual device A, 5% of people would choose to use it versus not. The gastric band, 11.6% people would choose to use it or not. And so this is a technique whereby you can mix the clinical data to come up with this probability or choice share to get a sense of what it would be like for these people, this population, when confronted with the opportunity to use the treatment, how many people would choose to use it. And in fact, this particular concept, that choice share, really was one of the elements that supported that CDRH decision. And you could apply it to the population as a whole or to subgroups with greater or lesser risk tolerance if you want to refine this. But the example I'll spend a little bit more time on is a different method. Next slide. So let me show you an example of a fake problem from an anticoagulant. So you're either preventing clots or you're causing bleeding, except in this case, you could see that the drug has caused, out of 10,000 patient years, 20 cardiovascular deaths with a wide confidence interval that intersects 0. So it's not significant. But between that and the non-fatal, non-intracranial hemorrhage major bleeding, you wonder, is this a good drug? Well, you can also see it prevents 110 ischemic strokes that are not fatal. So there's the question. I caused non-significantly 20 deaths to any cause, but I prevent 110 non-fatal ischemic strokes. Am I helping more than I'm hurting? That's a hard question, at least and hard to resolve without weights or preferences. So next slide. What I have here is an effects table. It's the two drugs, drug plus standard of care, placebo plus standard of care, and the risk difference, the same numbers that were plotted. Now, animate, Carl. I'm going to add the mean weight, which is sort of kind of correct, but they're just sort of mocked for the example. And now I'm doing a simplified approach for multi-criteria decision analysis. The full model MCDA approach is more rigorous. It involves things called value functions, but I'm going to do something simple and just multiply the weights by the risk differences. So if you can animate, please. The contribution then is that risk difference times the mean weight. So 1 times 20.4, same. The minus 20.4 times 0.23 is a much smaller number. Now animate one more time, please. Now what you do is add the contributions and you get the difference, in this case, in score, minus 15.9. Now that number actually has meaning. What this says is the difference between these treatments is the equivalent in the utility scale of 15.9 fewer deaths per 10,000 patient years. So that is what this model is doing for you. It's converting 6 different endpoints, all with their own level of importance, to a standardized scale. And in this case, I picked death, so you could have used any type of scale. So the between treatment difference on average is 15.9, favoring the study drug. Okay, do I trust that? Well, next slide. MCDA is not meant to look at a single number and you're done. You dissect the results. You do sanity tests. You ask, do I trust it? So the first thing you might do is look at those contributions and ask, do they make sense? So you can see here that minus 15.9 is created of the sum of the blue bars. And the key drivers are obviously CV death and non-fatal ischemic stroke, which makes sense and is kind of intuitive from this example. But it also tells me that if I want to understand benefit risk, that's where I should focus. The other endpoints are nice to know, but they aren't the real key drivers. Next slide. So now I want to understand the effect of uncertainty. So I have uncertainty in my clinical data, and I have uncertainty in my weights. So I actually can do a Monte Carlo and account for every single uncertain parameter. And instead of having a single point estimate of minus 15.9, I now have the distribution of the difference in scores on the left. And now anything above 0 favors the placebo arm. So I could do the graph on the right, a cumulative density function, see where I hit 0, and that gives me the probability that benefits outweigh risks of about 65%. So this is very useful. Now I know, yeah, benefits outweigh risks on average, but it's certainly not a slam dunk. That 65% I can use to decide whether I'm comfortable, say if I'm a regulator approving this, or if I'm a sponsor sending this to health authorities. Now there's a lot more you can do with this. Next slide. This is just the beginning of the type of sensitivity analysis. So this is called a tipping point analysis. What I do here is I take each weight and I vary it over some range, here plus minus 2 standard deviations from the weight. Now, you could see all the bars on bottom don't get anywhere near the 0, which is the tipping point where you'd switch from favoring the study drug to the placebo. But the top one, the weight for non-fatal ischemic stroke, that bar goes all the way to the right. In fact, it goes all the way to 20, which is extremely favorable for the placebo. So this tells me that there is only one weight that is sufficiently uncertain to cause the model to reach the tipping point, but that it can cause the model to reach the tipping point. And this tells me if I want to reduce my uncertainty, if I want to take that 64.8 or whatever percent and move it higher, I want to focus on reducing the uncertainty of that weight. Or it tells me this is the limit to how much I can trust this particular model with the data I have. Now, there are many other ways to do sensitivity analyses on a multi-criteria model. And technically, this is actually called the stochastic multi-attribute acceptability analysis type model, but they're really the same, just with a different degree of math. But I wanted you to get a sense that you can take this MCDA type model very far, and you could use it to dissect the results and decide whether you trust them, or if I'm going to invest resources in reducing my uncertainty, where do I invest them? Next slide. So my takeaway message is there are numerous approaches that you can use to apply patient preference data to inform the decisions. And I went through the 3 classes of preference data on its own, preference and clinical data looked at in parallel, or preference and clinical data mathematically combined into quantitative benefit risk analyses. The general guidance is to use the simplest approach that addresses your research question. So I often use a combination of approaches. And we are seeing an increasing interest by health authorities for these mathematical approaches. In general, in the past, they were more the realm for academia. But I and others have anecdotal experience where health authorities are asking to see some of this work. The final point I'll make is, as messy as some of them of what I might have shown you might look to some, the real-world application actually has another layer of complexity, where you have to do modeling for heterogeneity in your clinical data, heterogeneity in your preferences, dependency between your preferences, and dependencies between your clinical endpoints, nonlinearities in your preferences instead of that straight line or linear model, and many other issues. That all falls under the realm of mathematics, and it just tells you that you certainly can apply this work, but when it comes to real-world applications, sometimes you'll have to take it a step further. Anyway, thank you for the opportunity to share this thought on applying the preference data, the third part of the Prefer framework. We'll now switch to Arena Cleanput. We'll talk about HTA considerations for benefit risk for the preference studies.